Hey everyone, Simon here. Welcome back. Today, we're in Module 6 land again, genetic change, and we're zooming in on reproductive technologies.
And I'm Lachlan. Think of this episode as exam training for those describe, analyze, and assess questions about selective breeding and all the lab-style reproduction tricks humans do.
Let's kick off with a clear HSC-style define. Selective breeding, which is artificial selection, is when humans choose which male and which female get to reproduce because they've got desirable traits, like high milk yield or fast growth.
In exam language, that means we are changing the gene pool. The gene pool is the full set of alleles, versions of genes, in a population. If we only let the top animals breed, their alleles increase in frequency. Others drop out over time.
The classic cattle example. Cross a Frisian bull, famous for huge volumes of milk, with a Jersey cow, which makes really creamy milk. Some calves end up with alleles for both traits, so they produce large amounts of creamy milk.
Then the breeder selectively breeds those superstar calves again. So the alleles for lots of milk and creamy milk get more and more common in the herd's gene pool. That's literally altering allele frequencies with artificial selection, not natural selection.
But there's a catch. Some hybrids can inherit unwanted alleles at the same time. Those Frisian times Jersey cows can grow udders so large they struggle to walk and can get ulcers where the udder rubs against the legs. So productivity can go up, but animal welfare might go down.
And doing this with whole animals is slow and expensive. You've got to move big cattle around, hope they actually mate, and then wait for pregnancy. That's where artificial insemination, or AI, comes in. Let's describe the process, step by step.
All right, step 1. Collect semen from a chosen male, usually using mechanical stimulation or an artificial vagina. That semen contains millions of sperm.
Step 2. The semen is divided into tiny plastic semen straws, chilled and then frozen in liquid nitrogen. Picture a big tank full of colorful straws. That's literally a sperm bank for bulls.
Step 3. Transport and storage. Those frozen straws can be shipped around the world, much easier and safer than sending a massive bull on a plane.
Step 4. Insemination. When a cow is ready, the straw is thawed and loaded into an artificial insemination gun. The technician gently inserts the gun into the vagina up to the cervix and deposits the semen right where it needs to be. Not glamorous, but very effective.
Does that process make sense? Collect, freeze, ship, thaw, insert. That's your 4-mark describe artificial insemination answer right there.
Now, outcomes. For productivity, AI is huge. One bull can sire offspring in many herds and even long after he dies. Dutch bull Sonny Boy produced about 1.7 million units of semen in the 1990s. American bull Toy Story smashed it with 2.4 million units and around 500,000 daughters in 50 countries.
So, in exam terms, if they say, analyze the outcomes, you'd say, AI increases productivity and efficiency, more calves from the best males, cheaper transport, less injury risk during mating.
But it also reduces genetic diversity. If just a handful of superstar bulls father huge numbers of calves, their alleles dominate the gene pool, and other alleles are lost. The allele frequencies become skewed towards those few males.
AI isn't only about farming either. It's used in conservation. There's been work on sharks, artificial insemination in a broad-nosed 7-gill shark as a model, aiming to help endangered gray nurse sharks. And Missouri, the first gorilla born using AI, arrived at Melbourne Zoo back in 1984.
So if a question says, assess the impacts for conservation and genetic diversity, you'd balance it. AI can boost numbers of endangered species, but if we rely on a small number of donors, we still risk low genetic diversity and long-term health problems.
Nice. So basically, selective breeding plus AI lets humans drive artificial selection for our preferred traits. But we have to watch what that does to the gene pool and biodiversity.
All right, next concept, IVF and MOET. This sounds super high-tech, but the core idea is simple. Get lots of embryos from high-merit parents, especially cows.
Let's do a describe the steps run through. Lachlan, want to walk us through IVF first?
Sure. IVF is in vitro fertilization. In vitro meaning in glass. Step 1. Hormone stimulation. The female, say a cow, is given hormones so her ovaries release more eggs than normal.
Step 2. Egg collection. Those eggs are taken out of the ovary. In animals, this is often done by a vet using a needle guided by ultrasound.
Step 3. Fertilization in a petri dish. We mix the eggs with sperm from a chosen male in the lab. Fertilization happens outside the body, forming zygotes.
Step 4. Culture. Those zygotes are grown in an incubator until they reach an early embryo stage, a little ball of cells.
Step 5. Embryo transfer. A fresh or thawed embryo is put into a uterus, either the biological mother or a surrogate. That's done with a thin tube called a catheter that places the embryo into the uterine lining.
And step 6, which students sometimes forget, freezing. Extra embryos can be frozen in liquid nitrogen for later transfer or research. So that's your full describe IVF answer.
Now, MOET, multiple ovulation embryo transfer, often runs with IVF. Basically, cows are hormonally stimulated to ovulate multiple eggs. They're fertilized, and several embryos are produced from that one high-value cow in a single cycle.
Then those embryos are transferred into lots of surrogate mothers. A cow that would normally have one calf a year can now produce heaps of genetically related calves, even in different herds and countries.
So in explained terms, IVF and MOET massively increase reproductive output of a few selected animals. That shifts allele frequencies in the population towards the alleles carried by those elite parents.
Compare that to AI for a sec. AI spreads the alleles of one male widely, but each female still has one calf a year. IVF/MOET crank up both sides, lots of embryos from the same female and sometimes the same male too. It's artificial selection on steroids.
And contrast it with natural selection. In natural selection, alleles that increase fitness in the environment, survival and fertility, become more common. With IVF and MOET, humans select alleles that boost human goals, like milk yield, even if they don't improve survival in the wild.
So what are the consequences? Let's do an assess-style breakdown. First, genetic diversity. IVF produces many viable embryos from a small set of parents, so overall genetic diversity in the herd often decreases.
Second, infertility alleles. This is a sneaky one. IVF is often used when there's reduced fertility in one or both parents. Alleles that would normally be selected against because the animal can't reproduce naturally can now be passed on. Over generations, we might actually be breeding infertility into the population.
That's basically the opposite of natural selection, where alleles that improve fertility tend to increase in frequency. Does that idea click? We're helping individuals who would have been filtered out to pass on their genes.
Then we've got sperm banks for both animals and humans. They let people choose donors based on traits. In humans, maybe height, academic ability, or looks. That can increase the frequency of certain alleles and reduce others seen as unfavorable.
The risk is we might lose alleles that turn out to be important, like for creativity or disease resistance. So, from a long-term fitness angle, populations could become less resilient to new diseases or environmental change.
Ethically, you can also mention issues of designer traits, animal welfare in high-output breeds, and fairness in access to these technologies. Just keep it short in the exam and link it back to impacts on gene pools and fitness.
So if a question says, assess the consequences of IVF and MOET, you'd weigh the big positives: more offspring from the best animals, solving some infertility, conservation potential, against reduced genetic diversity, spread of infertility alleles, and possible lower long-term fitness. Let's switch to plants, artificial pollination, and hybridization. Same Module 6 ideas: gene pools, allele frequency, artificial versus natural selection.
Artificial pollination has been around for ages. There are records from ancient Assyria showing people hand-pollinating date palms, and Mendel used it in his pea experiments. The basic process is actually pretty simple.
If you had to outline the process in an exam, you could say, Humans remove the stamens, the male parts, from a flower, collect pollen, then dust that pollen onto the stigma of a flower. That can be on the same plant, which is self-pollination, or a different plant of the same species, which is cross-pollination.
Doing it by hand gives breeders control over which parents cross. They can combine alleles for things like disease resistance or bigger fruit. That's artificial selection again. Humans decide which alleles enter the next generation.
Outcomes-wise, artificial pollination is often used to create hybrids. Maize, corn hybrids are famous. Higher growth rate, greater uniformity, increased yield.
And in Australia, wheat breeders crossed a purple straw variety called 14A with a Fife Indian variety, Yandilla, to create a new hybrid called Federation. New combinations of alleles entered the wheat gene pool, and because those hybrids were fertile, those combinations increased in frequency.
So unlike some animal techs that shrink diversity, hybridization within a species can actually increase genetic variability because you're mixing different lines. New allele combos appear in the population.
But there's a twist. If farmers worldwide all choose the same few high-yield hybrids, biodiversity drops at the larger scale. Crops become too genetically similar.
The 1970 U.S. corn blight is the classic cautionary tale. A fungal disease ripped through corn that all shared similar genetics. And because of that low diversity, huge areas of crops were wiped out. So more yield, but more vulnerability.
We also rely heavily on insect pollinators. Researchers compared gooseberry plants pollinated by insects to those pollinated by hand. The insect-pollinated plants had larger fruit and higher seed germination rates, so natural pollination sometimes produces fitter offspring than our artificial attempts.
And as bee numbers drop, scientists are exploring drone pollinators, little flying robots with sticky surfaces that carry pollen between flowers. It's wild, but again, it's humans taking control of reproduction.
All right, let's finish like an HSC compare question. Compare selective breeding with AI, IVF or MUAT, and artificial pollination in terms of process and impact on genetic diversity.
Process first. Selective breeding is the broad idea. Choose parents with desirable phenotypes and let them mate. AI is more specific. Collect sperm, freeze in straws, and deposit it into females. IVF/MOAT take fertilization and early development into the lab, making mini embryos from elite parents.
Artificial pollination is the plant version. Manually move pollen between flowers to control which plants cross, often to create hybrids.
Now, impact on allele frequency and genetic diversity. All four are artificial selection. Humans decide which alleles are passed on. In selective breeding, AI, and IVF/MOAT, we usually reduce genetic diversity because we reuse a small number of high-performance animals, so their alleles dominate the gene pool.
IVF or MOET especially can spread infertility alleles and reduce long-term fitness, even while short-term productivity rises. Sperm banks can push certain donor alleles to higher frequency and risk losing others.
Artificial pollination and hybridization can increase genetic variability within a species by creating new allele combinations, like in Federation wheat or modern maize hybrids. But heavy reliance on just a few hybrid varieties reduces biodiversity across farms and makes crops more vulnerable, like the U.S. corn blight showed.
So if you're tying this back to Module 6, you'd say, these reproductive technologies are powerful examples of humans altering gene pools and allele frequencies through artificial selection, sometimes opposing what natural selection would do in terms of fitness and fertility.
Does that feel a bit clearer? Think, who is choosing the alleles, nature or humans, and what happens to diversity and long-term survival?
If you can answer that for each technology, you're in great shape for those compare and assess questions in the HSC.
All right, we'll leave it there for today. Emma, thanks for walking through all the techie bits.
Anytime. Good luck with your revision, everyone. Go back, pause, and jot down those key definitions.
We'll catch you next time for more Module 6 goodness. Bye.
See you later.